So my talk's going to be a little bit different than the preceding talk. So my research per se is not driven by evolution. So my lab works in bacterial pathogenesis. However, the organism that I work on, chlamydia, is an obligate intracellular pathogen. So it's really been driven by these reductive evolutionary processes. So it's an endosymbiont. So what I'm going to do today is tell you a story about endosymbionts. Okay, and when I was looking at the other talk titles and what everybody else is doing, I realized that it's really my job to bring you the small today. So we're going to be talking about microbiology and microorganisms. So what's an endosymbiont? Right? So this really comes from two Greek terms, and the concept is you have organisms living together, and these organisms, you have one living inside of the other one. So this goes back to that whole nesting doll concept, though it would be unfortunate to be the one that's stuck inside of the Wookiee. So just an example of what I'm talking about when I'm talking about these endosymbiotic relationships. So this organism that you see here, this is a paramecium. So this is a single-celled eukaryotic organism. And then all these green things that you see stuffed inside of this organism, this is an algae. Okay, so this is an example of a eukaryotic organism living inside of another eukaryotic organism. And these algae are chlorella. And this relationship is a mutualistic relationship. So the algae provide nutrients to its host paramecium cell, and the paramecium provides things to the chlorella. Okay, so there's an endosymbiotic relationship where both partners are benefiting. Okay, so we're going to define an endosymbiotic relationship as a relationship where you have a single-celled organism living inside of a single host cell. And you can break this down into different groups. So you can look at lifestyle. Okay, so if you're a facultative endosymbiont, such as the chlorella, which I just showed you, it means that you can live free in the environment. However, if you're taken up into a host cell, you can do just fine in that host cell, assuming that you've entered into the correct partnership. And you also have obligate relationships. So the organism I work with, chlamydia, is an obligate endosymbiont. And these organisms have lost the ability to survive in the environment, and now they can only exist and replicate and grow inside of a host cell. And you can take those relationships one step further, and you can look at who's benefiting. So the idea is you can have a parasitic relationship. And we're talking about benefits. We're usually looking at it from the endosymbiotic standpoint. So the endosymbiont is parasitizing the host cell. Okay, so it's basically succeeding at the detriment to the host cell. You can have a mutual relationship, and that's what we just showed you with chlorella, where both organisms are feeding each other. Then you can have a neutral relationship. And basically, when we look at neutral relationships, we're saying nobody benefits. But my understanding of those situations is really that we just don't completely grasp how these organisms are really interacting. And I think you generally go in one direction or the other. So I just want to start with some examples of facultative endosymbionts. So there's a whole collection of human pathogens that fall into this category. This list is by no means exhaustive. So you have a number of bacteria, fungi, as well as protozoa. Within the bacteria, you may have heard more recently about Listeria monocytogenes. This is a foodborne pathogen, so that was associated with some of the deaths that came from people eating contaminated cantaloupe. So Listeria can live in the environment. It can colonize food. And then when you consume it, what it actually does is your intestines is it will invade into your epithelial cells. Okay, so it's a facultative interaction. It can live on its own, but it's perfectly adept at getting into host cells, replicating, and then going on and damaging those cells and moving on. So this is a parasitic relationship. Listeria benefits, the host cell does not. And you have a number of facultative endosymbionts that actually reside. A very common theme is we see these residing in insects. Okay, so these are ones that, again, can live in the environment, but when they get into a host cell, they do just fine. And these are mutualistic relationships, so generally both partners benefit. Okay, but I want to spend a little bit more time talking about my favorite endosymbionts, which are your obligate endosymbionts. Okay, so much like Blanche Dubois from Streetcar Named Desire, they become dependent upon the kindness of strangers. In this case, the stranger is going to be a host cell. So again, you have a number of human pathogens that fall into this category. My favorite is chlamydia, which is an obligate intracellular gram-negative pathogen. This is an example of chlamydia here. So this would be a host cell. And we work with a lot of epithelial cell lines in labs. So these are cancer cell lines. So you often have multiple cells fused together. So a eukaryotic cell would normally have one nucleus. This is something we call a giant cell. So it's actually carrying around 3 nuclei, which you see here in blue. And this green staining you see here, this is essentially a bag of chlamydia. Okay? So you probably have 500 to 1,000 bacteria here living inside of this cell. So after about 2 days' time, they're going to lyse this cell and go on and find another host cell to go in and grow and then keep their process going. So this is a parasitic relationship. Within your obligate endosymbionts, you also have a number that enter into either mutualistic or neutral relationships. Okay, so when I'm talking about these relationships, I'm really talking about shared nutrients. Okay, so the bacterium is providing something to the host cell that the host cell needs, and the host cell is typically providing something to the bacterium that the bacterium needs. So don't get lost in everything that's going on here. What I want you to focus on is the organism here. This is Buchnera. Buchnera is an obligate endosymbiont that lives in cells from aphids. And in green here is the cytoplasm of the aphid eukaryotic cell that it's living inside of. And the concept for a mutualistic relationship is that the host cell is providing things to the bacterium, and the bacterium is providing things to the host cell. If this was a parasitic relationship, it would be a one-way street, where basically the host cell is providing things to the bacterium's not sending anything helpful out, and actually what they send out are things that end up killing the host cell. Okay, so how do you end up as an obligate endosymbiont? Okay, and this is going to get into the little evolutionary portion of our talk for today. So if you look at a bacterial genome, okay, and you think about a bacterial genome can expand and a bacterial genome can collapse. And we move towards the obligate path, we're talking about genome collapse. So the concept is you can grow your genome through gene acquisition. So your bacterium can pick up DNA for the environment. It's a very common process. Take it in, and they can either chew it up as energy, or what happens sometimes is they'll incorporate some of this into their genome, so they'll expand their capacity. They can also duplicate their genes, so they can take an existing gene, they can duplicate it, and then that duplicated copy can become something new, so it can diverge off, but you're expanding your genome. When we talk about the movement from a free organism to something that's an obligate, we're really talking about the concept of gene loss. Okay, so basically you're losing the ability to live on your own, and you're getting trapped inside of a stable environment. Okay, so I just want to give you an example of how something like that can happen. So what we're looking at here would be the genome of a bacterium. Okay, and what we're looking at in red here is a gene known as BioY. This makes a protein which allows a bacterium to obtain biotin from the environment and take it up. So biotin is an essential nutrient for bacteria as well as for us. The genes shown here in red are genes that allow a bacterium to synthesize biotin. Okay, so this bacterium can either make biotin on its own, or it can steal it from the environment. So if you think about if you're out in the environment, if there's a source of biotin from an energy standpoint, it makes more sense to grab it than to synthesize it. So this bacterium can do either. When it enters into a relationship with a host cell, so if you can think about a free-living bacterium figures out a way to get into a host cell. So it becomes a facultative endosymbiont, which can transition between these two places. Inside of a host cell, it's a pretty stable environment. Okay, once you can figure out how to keep the host cell from killing you, it's actually a big bag of goodies. Okay, and there's a lot of biotin in there. So they're going to turn on their biotin transporter. They're going to shut down their synthetic pathway. Okay, and in this context, there's really no selective pressure to maintain the ability to synthesize biotin. So what happens is, as these organisms divide, you can have an error resulting in a mutation in your biotin synthesis genes. So you end up with mutant bacteria that can now still transport biotin, but they're no longer able to synthesize. So what happens is, this bacterium was a facultative endosymbiont. Okay, so it's going to do its thing, and it's going to eventually leave the cell. When your mutant goes back out into the environment, okay, if there's not biotin in the environment, it can no longer synthesize it. Remember, it picked up these mutations. So essentially, it's going to be selected against. Okay, so now these mutant bacteria can only survive if they maintain their niche within the host cell. Okay, whereas the wild type can continue to transit back and forth. So what happens is, over time, these mutants start to pick up other mutations and other pathways, and essentially their genome starts to shrink down. Okay, so you might imagine, right, if you looked at the average genome size for a free-living or facultative bacterium, we would hypothesize it would be larger than what you see for obligate endosymbionts. Okay, so we view this as a pathway to dependency. That's exactly what we see. So looking here is genome size in million base pairs. So this would be a collection of free-living and facultative bacteria from what have been sequenced. And if you look at the average genome size, it's about 4 million base pairs. Okay, and you can make a rough correlation to the bigger your genome, the more capacity you have. And if you think about it from an environmental standpoint, the more niches where you're going to be successful. As we move to the obligate, remember we talked about how gene loss can contribute to an obligate nature. If you look at genomes from obligate endosymbionts, okay, it's about 1 million base pairs. So you're seeing a loss of about 3 million base pairs, or about a 75% loss of gene capacity. Okay, so gene loss contributes to your obligate status. Okay, so I want to tell a neat story on how basic science, okay, and understanding endosymbiotic relationships has really been translated to something that's now being used to help human health. So this story is with Wolbachia, which was discovered by Bert Wolbach in the mid-1920s. This is an insect cell, and inside of these cells, this is Wolbachia. So Wolbachia is a gram-negative bacterium. So Wolbachia infects various arthropods, and what's going to be key to this story is we can make Wolbachia infect mosquitoes. So most mosquitoes are generally Wolbachia-free, and there's going to be an important consequence of us being able to get Wolbachia to grow in mosquitoes. So Wolbachia is transmitted vertically, so it resides within the reproductive tract of infected insects. So when you transmit, if you think of this as an infection, you can have horizontal transmission, which is somebody has the flu and they sneeze and you get it, that's horizontal. Vertical is from the mother to the offspring, okay? So if we look at fruit flies, which is where the initial work where Wolbachia was done and a lot of the basic science that gave us the understanding of how Wolbachia works. So you can have infected male and female fruit flies. It's only transmitted by the female. Okay, and within the female, it lives in the regions that are responsible for egg production. Okay, so we don't have to understand everything that's going on here. The key is this is your late stage where you're producing your eggs. Everything you see here in green, this is your Wolbachia. So Wolbachia is associated with the egg. So when that egg is fertilized and you give rise to offspring, those offspring are going to be infected by Wolbachia. So they started doing a bunch of mating experiments, okay, and that's one way they figured out it was transmitted vertically. But what they found is if you take an infected female, an infected male, all your offspring are infected, right? Not too surprising. Uninfected partners, you have uninfected offspring. If you have an infected female and she mates with an uninfected male, okay, all your offspring are going to be infected. It's coming from the mother. However, if you take an infected male and you mate that male with an uninfected female, you end up with no offspring. Okay, so basically the embryos are aborted. So this is something known as cytoplasmic incompatibility. Okay, and the mechanism behind this is still being worked out. But the concept is, again, if you have an infected male and you would release this male in a fruit fly population, every uninfected female that he mates with will give rise to no progeny. Okay, so people started thinking about this and how they could make this applied to reduce certain insect vectors that carry around human diseases. So I want to hit on 2 human diseases. Dengue fever, this is caused by a virus, infects primarily in tropical regions, so you have about 50 to 100 million cases each year. And malaria. Malaria is caused by Plasmodium, which is a protozoan. Again, infects in tropical regions. Okay, high rates of morbidity and mortality. What both of these have in common is they're both transmitted by mosquitoes. Okay, so people hit upon this concept of can we use Wolbachia as a biological control mechanism to decrease mosquito populations, right? If you get rid of mosquitoes, which carry around these vectors, you're going to decrease your disease incidence. So the concept went back to cytoplasmic incompatibility. So here are your wild-type mosquitoes. Okay, you have a female here, and females are the ones that actually transmit the infection. Here you have an uninfected mosquito. So right now, when infected and uninfected, we're talking about carrying dengue virus. And you have an uninfected male. So every time males and females mate, they make more mosquitoes. So the idea is if you can get a male line of mosquitoes that's infected with Wolbachia, and you release those male mosquitoes into the population, every time these male mosquitoes mate with a female mosquito, the female mosquitoes aren't carrying Wolbachia, you're not going to have any progeny. And the concept is over time, your mosquito population drops. Okay, one of the other interesting things that came out about studying Wolbachia infections in insects, okay, and this holds for Drosophila as well as with mosquitoes, is that your infected mosquitoes are also resistant to some of the pathogens that are spread from vectorial bites. Okay, so they're less likely to pick up dengue, they're less likely to pick up Plasmodium and some of these other vector-borne pathogens. Okay, so when you see these kind of headlines, right, so Wolbachia infected mosquitoes released in Vietnam with hopes of stopping dengue, and so on, this is what they're talking about. Okay, and a lot of it goes back to this the condition of cytoplasmic incompatibility, but using this as a biological control mechanism. But I think it's a really nice example of how basic science was taken and applied translationally for human health. So just some key points coming out of the talk that I want you to walk away with. Again, our definition of a single-celled organism living inside of a host cell. The relationships are complex, okay, but we can define those relationships in two primary manners. The lifestyle, so whether it's a facultative relationship like we talked about with chlorella, where it can live in the environment but does just fine in the right host cell, or an obligate relationship where they've lost the ability to live outside, and a lot of that has to do with reductive evolution and loss of their genome structure. And you can look at benefits. So is it a parasitic relationship? Is it mutual? Or is it neutral? These relationships involve a lot of direct communication. So I showed you that figure of all those metabolic pathways and things coming in and coming out. And the idea is you have a defect in the metabolism of the host cell, and you have a defect in the metabolism of the bacteria, but they don't overlap. So they can make up for each other's shortcomings by sharing nutrients with each other. And the last concept that I want to leave you again with is the issue of gene loss, and gene loss leading to an obligate lifestyle. And one thing that I decided to pull from the talk, because I wasn't sure if we would have time, is the endosymbiotic theory for the origin of mitochondria and chloroplast. Okay, and that was mentioned briefly in the first talk, but the basic concept is your mitochondria have a genome, chloroplasts have a genome, and one of the things people have been trying to figure out for a long time is how that was possible. And basically what we now believe is that you had an organism that entered into a facultative relationship that eventually became an obligate, and through a series of genome reduction events became something that's no longer its own organism and something we now consider to be an organelle. Okay, so if you're interested in this, and these are going to be up on YouTube, if you go in to evolution.berkeley.edu and you look up endosymbiosis, they'll give you a real nice story on the endosymbiotic theory of origin for your mitochondria and chloroplast. So I'll just leave with the nesting doll analogy, again going back to something that I think Darwin might be familiar with. And thank you for your time, and I'll be around at the end for any questions.